Okay, everyone. We'll get started. So today's talk is from Simona Infantino. So Simona's a postdoc in David Tarlington's lab. And Simona's really interested in all things related to signaling in lymphocytes, particularly in B cells. And I guess she got that interest during a postdoc with Mikhail Rett in Freiburg at the Max Planck Institute of Immunobiology, where that lab is one of the world leaders in understanding B cell receptor signaling. And during that postdoc, Simona made, I think, a serendipitous finding of an unusual modification to components of the B cell receptor, and figured out what that was, and it turned out to be, as the title suggests, Arginine methylation, which at least to us was a new type of post-translational modification. And then when she came to David's lab, she decided to introduce some genetics into this question and try to ask what happens when you remove the enzymes that make this particular modification. And that's what she'll talk to us about today. So after you, Simona.
Thank you. Okay. Thank you, Steve, for the kind introduction. Today I will talk about my favorite post-translational modification, as Steve said, and this modification is arginine methylation, and in particular I will talk about the impact that arginine methylation has on B cells and their receptors. So the enzyme that methylated arginine are called the protein arginine methyltransferases. Protein arginine methyltransferases catalyze the transfer of a methyl group from S-adenosyl methionine, that is the methyl donor, to arginine, to the nitrogen group of an arginine. And specifically, this modification, this process produces three different products that are depicted here: monomethylated arginine and demethylated arginine. There are two major types of protein arginine methyltransferases, and both types can produce monomethylated arginine. While type 1 can induce asymmetric demethylation, that means both methyl groups are deposited on one of the available nitrogen group on arginine. Type 2 PRMTs can instead provide symmetric demethylations. That means that the two methyl groups are deposited in the two different available nitrogen groups of the arginine. Up to now, there are about 9 enzymes that have been characterized, and I depicted here the most popular one. And as you can see, I highlighted in red the PRMT1, that is the methyltransferases I will talk about today. And as my title said, I will mainly talk about this post-translational modification in a B cell, so in a subset of lymphocytes. However, I just want to remind you that arginine methylation is a broad post-translational modification, and in this table there are examples of proteins that are not involved in B cell receptor signaling, but they are indeed methylated. As you can see, you have chromatin-associated protein as well as splicing protein, and also DNA damage response protein have been found to be methylated, as well as viral protein and signaling proteins. So let me introduce you the enzyme, so PRMT1. PRMT1 was the first protein arginine methyltransferases that have been identified in mammals, and this happened about 20 years ago. It's been shown that PRMT1 account for about 85% of the overall arginine methylation in rat fibroblasts as well as in mouse liver cells. And the enzyme has a wide variety of substrate specificity. However, it seems to prefer arginine that are flanked by one or more glycine. Because PRMT1 is expressed in a broad variety of cells, the knockout mice are embryonic lethal. Indeed, the mice cannot develop, they simply can't develop, and they die at about embryonic stage 6.5. Luckily, very recently, a group from Canada, the group of Stephen Richard, had generated conditional knockout of PRMT1. And in this paper, they had mainly characterized the role of PRMT1 in mouse embryonic fibroblast. And in particular, they've shown that when mouse embryonic fibroblast lack PRMT1, they go through a spontaneous DNA damage, they have defect in cell cycle progression and in checkpoint, and they do present aneuploidy and polyploidy. Now, what's the connection between arginine methylation and B cells? So before entering into those details, I just want to give you a brief introduction about the BCR receptor. As you can see from here, BCR receptor is composed by the immunoglobulin chain and the signaling subunit Ig alpha and Ig beta. Upon antigen encounter, there is the activation of a SAC family kinase lin, and that induced phosphorylation within the cytoplasmic tail of Ig alpha and Ig beta, specifically those two tyrosine that are present in the ITAM motif, that is the immunotyrosine activation motif present in those cytoplasmic tail. And this event allowed the recruitment of another important kinase in BCR receptor, that is SYK. This kinase then phosphorylate the adaptor protein SLP65, and this generate all the signaling complex that are important for the calcium signaling, cytoskeletal reorganization, and eventually alter gene expression pattern. So as Steve said, my interest in arginine methylation came from my previous lab when I was working in the lab of Michael Red, that, as Steve mentioned, is very famous for his BCR receptor studies, and indeed is also the person that identified the ITAM in the Ig alpha tail. When I was in his lab, I was mainly focusing my interest in studying the Ig alpha signaling subunit of the BCR receptor. And by doing one experiment, so we had in the lab an antibody that was not what that rumor says that wasn't a really good antibody, and these antibody were raised against the cytoplasmic Ig alpha tail. However, as you can see here from indicated in the red box, so the recognition is very good in unstimulated B cells. However, upon certain stimuli, the tail of Ig alpha was no longer recognized. So, of course, when I saw these results, I was a bit disappointed because it was my loading control, as you can imagine. But after thinking a bit more, I arrived to the idea that maybe the recognition was prevented by some post-translational modification that was occurring in the Ig alpha cytoplasmic tail. And indeed, here is just part of the sequence of the Ig alpha cytoplasmic tail. I have noticed that in the tail was present an arginine, and this arginine was highly conserved between mammal species. And very interesting, this arginine was also flanked by one glycine. And as I told you before, the preferred substrate of PRMT1 are arginine that are flanked by one or more glycine. So I wanted then to test this hypothesis if indeed Ig alpha was methylated. And I was at the right time in the proper place because Max Planck Institute was recruiting many epigenetic groups. So I did then, I could establish then a collaboration with one of the epigenetic group that joined the institute, the group of Robert Schneider, and then I could test my hypothesis. And indeed, yes, I found that Ig alpha was methylated. Here is just a summary of the first experiment we had done, and it's a very simple experiment. And we took advantage of an in vitro system, and we use EC293 cells. We did overexpress different PRMTs available in those cells, and all the PRMTs were flanked at the N-terminus by an HA tag that make very easy for us to immunoprecipitate it. And then all the immunoprecipitated PRMTs were put together in an in vitro methylation assay with a recombinant protein that was generated by GST and cytoplasmic tailor of Ig-alpha, as well as the control, in the presence, of course, of a radioactive methyl donor, that is S-adenosyl methionine, and the methylation was then developed by autoradiography. Very briefly, as you can see from this red box, we were able to discover that Ig-alpha was indeed methylated, and this methylation was mediated only by PRMT1. Here is just a control to show you that all the PRMTs that have been tested, and we tested more than what I'm showing you today, but this is just for simplicity, they were indeed active and they were able to methylate H2A. After that, we did prove with another experiment that the only methyltransferases that was able to methylate Ig-alpha was indeed PRMT1. Again, we use an in vitro system, so we overexpress this time Ig-alpha in EC cells, and we knock down PRMT1 with small interfering RNA or using the relative control. We lyse the cells, and at this time, we use specific antibody against the methylated arginine on Ig-alpha. So you have to think that from this slide to the previous one, it's about 1 year time frame. So as you can see from here, when you knock down PRMT1 and the efficient was very good, you also abolish arginine methylation. We then also found, but I won't show you today, that PRMT1 was a negative regulator in B-cell receptor signaling and was playing a role during B-cell development. Indeed, if you, we tested the expression of the enzyme in different B-cell subsets. As depicted here, the pre-B cells, so precursor B cell, express very high level of PRMT1, and they retain the transitional B cell present in the spleen. They are immature B cells. They also retain the level expression of PRMT1, and PRMT1 is also expressed in the more mature B cell stage, so the marginal zone and follicular B cells. It's not equally expressed, but is expressed in all subsets. So then I came to Australia, and I did convince my boss to bring here the mice. And so the mice were generated, as I told you before, from the group of Stephen Richard, and this is the way they've been generated. So they have the two LOXP side that flank exon 4 and 5, and by expressing a CRE, you just generate a frame shift and so a null allele for the PRMTs. Because my study and my interest in B cells, and also the same for David Tarlington, we decided to cross the PRMT1 flox mice with a CD23 CRE. So this CRE recombinase is under the control of the CD23 regulatory elements, so thereby restricting the expression only in the mature B cells. What we had found, so we had analyzed the mice, and we had found that deleting PRMT1 in the mature B cells does leave the B cell compartment intact. As you can see from here, on the top is wild-type control, but I just want to you to keep in mind that when I talk about wild-type, those mice also express the CD23 CRE, because we wanted to make sure that the phenotype that was seen was due to the lack of PRMT1 in B cell rather than CRE toxicity. So even if it's not indicated, they always express the CD23 CRE. And so this is, if you now get on a CD19 positive cell, that is a B cell marker, and the mature B cell are negative for CD93, And you can distinguish by 21 and 23 follicular and marginal zone B cell. And as you can see, the proportion of those cells are equally distributed between wild type and knockout, conditional knockout mice. And this is just a quantification of the absolute cell number of splenocytes as well as B cells in the spleen. As you can see, there's no differences. So we then decided to study in vitro the function of PRMT1 in B cells. So what we have done next, we isolated B cells from the spleen and of wild type and knockout mice, and we did activate, the first activation stimuli that we've given to the cells is the mitogen CD40 ligand in the presence of IL-4 and IL-5. So these stimuli allowed robust proliferation of the cells and induce, after a few days, the differentiation into antibody-secreting cells. So for this first experiment, the cells have been activated for about 2 days. And as you can see from the left panel, these are the wild-type B cells. Unstimulated B cell express a certain level of PRMT1 protein. However, upon stimulation, the PRMT1 expression gets upregulated. And of course, this is the control. The PRMT1 knockout B cell don't express PRMT1. This is kind of reensuring that the deletion had happened properly. We then had measured the level of overall arginine methylation in the same condition. Again, you can see that unstimulated B cells, wild type as well as knockout, have very few protein that are methylated. And upon activation, the methylation gets upregulated again. This is somehow mirroring the upregulation of the enzyme. And here I depicted with the asterisks some of the bands that are present in the wild type but not in the knockout B cells, suggesting that those proteins might be specific substrate of PRMT1. So this first experiment has been done isolating B cells from the spleen, so in mice. We then asked ourselves if also this was true for human B cells. We then isolated human B cell from blood donor, and indeed we had found that PRMT1 also upon activation is upregulated in human B cells. And I haven't shown you here, but the same is true for arginine methylation in human. Another important experiment that suggests how the arginine methylation machine is regulated in B cell upon activation have been done using the same stimuli as I told you before, so CD40 ligand L4 IL-5, again for a couple of days. And importantly, this time what we had performed was an ex vivo arginine methylation assay. So the cells were labeled and incubated for about 3 hours in the presence of radioactive methylmethionine. So the methylmethionine can get uptaken from the cells and can be converted inside the cells into S-adenosylmethionine. That is, as I told you before, the methyl donor. But of course, as you can imagine, if you label the cells with methionine, the same methionine can be incorporated in the newly synthesized protein. So to distinguish between these two processes, so arginine methylation and newly synthesized protein, we had do this assay in the presence of protein synthesis inhibitor. The cells were then lysis, and the arginine methylation was then developed by autoradiography. As you can see from here, in unstimulated cells, there is nothing. So in principle, what this experiment is telling us is that the arginine methylation machine is silent in naive B cells. So as I showed you before, there are proteins that are methylated in naive B cells. However, those proteins are probably have been methylated previously. And in activated B cells, the machine is very active. And as you can see from the numerous band from this autoradiography. And again, with the asterisk, I indicated some of the band that are present in the wild type but absent in the knockout B cell, suggesting possibly newly PRMT1 specific substrate. Again, we repeat the same experiment, isolating B cells and activating them with the same stimuli, but this time we wanted to know the role of PRMT1 in proliferation and differentiation into antibody-secreting cells. So to then track cell division, we label the cells with a cell dye, CTV, that gets diluted upon cell division. And we did monitor the cell number as well as proliferation at different time points, 2, 3, and 4 days after activation. As you can see from here, so this is on the x-axis, you have the CTV that gets diluted upon proliferation. And on the y-axis, you have CD138, that is a marker that gets upregulated into antibody-secreting cells. So this should represent our plasmablast formation after 4 days of stimulation. And as you can already see, there are very few cells that lack PRMT1 that could differentiate into antibody-secreting cells. Moreover, if you see the overall number of division that the cells have been going through, so you can notice that the PRMT1 knockout B cells had only gone through 4 divisions. And indeed, in all the experiments I've been doing, the cells stop to proliferate after about 4 divisions, while the wild type can proliferate up to 7 times, and also because after that you can't monitor any more proliferation. And this is just a quantification of the proportion of plasma cells, and as you can see, the difference between wild type and knockout B cell is significant. But we also had measured the absolute cell number of cells and compared the numbers between wild type and knockout B cells at different days. And as you can see, the knockout B cells are much less in numbers if compared with wild type at any time points. And this difference is significant. We then want to understand more about proliferation and survival and the role that PRMT1 could play in those processes. So we did repeat the same experiment, but this time we activate the cells with anti-IgM, so we trigger B-cell receptor. And again, the cells were labeled with CTV, and we did measure proliferation and survival of those cells at different time points. As you can see, specifically at day 4, where the difference is broader, so in the black line is the wild type and the gray line is the knockout, you can immediately notice that you have less cells that are knockout if compared to wild type, and also the cells have been going through less division number. And this is the quantification. As you can see already, 2 days after activation, the knockout B cells are less in number. And just want to point you the attention to the fact that at day 2, they have not gone through any division yet with these stimuli, suggesting that PRMT1 might play a role in cell death. And indeed, we had performed an experiment to see if we could rescue the survival of those cells. And we had used a pan-caspase inhibitor, QVD. So we repeat the same experiment, but this time we measure the cell number after 1 day, because already at day 2, the difference in terms of cell number was too big between wild type and knockout B cells. And of course, you have to remember that as soon as you trigger B-cell receptor, also wild-type B-cell die very quickly. However, the difference in cell number is still significant between wild-type and knockout. And by incubating the cells with QVD, we could rescue the survival of the knockout B-cells as well as of the wild type. And at this stage, the difference in cell number was not anymore significant, suggesting that PRMT1 might really play a role in a mechanism that is caspase dependent. So, and very recently, just a few days ago, we did measure the level of all the pro-survival molecule upon this activation. So the same stimuli as I showed you before, B cell were activated with anti-IgM, so we trigger B cell receptor for about 24 hours, and then we measure all these molecules, specifically MCL1, BCL2, A1. I haven't had the time to measure other molecules, so that's on the list to do next. So, but what you can see already, so in unstimulated B cells, so if you compare wild type versus PRMT1 knockout B cell, the level of MCL1 is very similar. However, upon activation, you can clearly see that MCL1 is not retained in knockout B cells, so it's really, really low. And the same was true for A1 and BCL2. The difference in A1 is not expressed in unstimulated B cells, but is upregulated upon activation. This really suggesting that maybe there's a role of PRMT1 in regulating all these pro-survival molecules. So to conclude my first part, what I show you until now is that PRMT1 is upregulated in B cells, and I just want to remind you this wasn't known until now, and I show you that PRMT1 knockout B cells have defects in proliferation, survival, as well as differentiation. And apparently, there is a defective regulation in MCL1, A1, and BCL2 in activated B cells that lack PRMT1. Okay, then now, in vivo veritas, that. So what I show you so far is all done in vitro, but what's happening in vivo? Just here, I depicted a secondary lymphoid organ, as I just want to remind you what happened upon during an immune response. So B cells, upon activation, they migrate at the edge between the B and the T cell area to encounter and interact with the cognate T cells. This event induces a robust proliferation, and the cells differentiate initially into plasmablast and early memory B cell, and then most of the cell migrate to the center of the follicle to then give rise to germinal center reaction, so as depicted here, germinal center B cells, and then they will further differentiate into long-lived plasma cells and memory B cells. So before going into the in vivo details, I just want to show you the expression level of PRMT1 in these three subsets. So this is what I call non-GC, that in principle are the naive B cells, so the follicular B cell I showed you before. And I compared the expression of PRMT1 that you can't see, of course, between these naive B cells and germinal center B cells and antibody-secreting cells. So as you can see, you don't see PRMT1 in the naive B cell because I didn't overexpose, but if you expose a bit longer, you can see the level. So it's clearly upregulated in germinal center B cells that represent activated B cells, and it's also retained in the more differentiated subset antibody-secreting cells. And this is kind of important for what I will show you next. So what we did next was to immunize mice with a protein antigen and PKLH in alum. And this immunization induced robust proliferation that peaked around 7 to 10 days upon injection. And in this experiment, we had measured the antigen-driven B-cell expansion after 7 days of delivery of our antigen. And this is the spleen. On the top panel, you have the wild type. This is gated on the B220 positive cells, that is a B-cell marker, and negative for IgM, IgD, and GR1. So if you get on those population, you can distinguish the antigen, so MP positive versus IgG1. This is the box you have to care about. So these are the activated antigen B cells, specific B cells. And this is the wild type. You can see they had responded properly. However, this population was completely missing in the mice that had deleted PRMT1 specifically in B cells, suggesting that PRMT1 was required for the antigen-driven B cell expansion. We next check the germinal center, and again, as you can see, the wild type, they had generated appropriate germinal center. However, this population was missing in the knockout mice. And also visually, this is an histology in which you can see that GL7 is a germinal center marker in orange, and IgD that mark all the follicles. You can see inside the follicles the foci of the germinal center very nicely in the wild type, but those foci are completely missing in the knockout mice. And this is just a quantification of the absolute cell number of germinal center B cell that we have found, and the difference is significant. And as I told you before, after activation, also the cell differentiate into antibody-secreting cells. So we did also measure the level of antibody-secreting cell upon immunization. And these panels show we had done that by ELISpot. As you can see from this panel, we did plate 10 to the 6th or 10 to the 5th cells from the spleen. The top part is the wild type, and the bottom is knockout, and you can already see visually the difference in terms of antibody-secreting cells. So I just want to remind you that each dot should represent one cell, so one antibody-secreting cells. And this is just a quantification that PRMT1 was absolutely crucial for the generation of the MP-specific antibody-secreting cells. And somehow what I show you so far has fit perfectly with the expression of the enzyme in the different B cell subset that is is very highly expressed in germinal center as well as in antibody-secreting cells. However, there are instances in which mice cannot respond properly to a protein antigen, but they can mount a proper immune response upon infection. So to make sure that, to test this hypothesis, we did infect the mice with influenza virus, and we did test the immune response 7 days after intranasal infection. And as you can see, again, the top part is the wild type and the bottom is the knockout. The wild type mice had mount a proper germinal center, so they generate germinal center. However, this population again was missing in the knockout mice. And the same was true for the antibody-secreting cells. This is CD138 versus B220, and this is the antibody-secreting cells population. However, I just want to point out the fact that the mice, the knockout mice, had responded to influenza. It's just the B cell compartment that was defective. And this is shown here by the CD8 positive, tetramer positive, double positive cells here that are equally represented between wild type and knockout. We have then analyzed, so what I show you so far is a T-cell dependent immune response, but we wanted also to know if PRMT1 was important to mount a proper T-independent immune response. So to do so, we had immunized the mice with MP Ficol and had measured the 5 days after immunization the level of activated B cells and the antibody-secreting cells. As you can see from here, so the wild type had responded properly, and this time this kind of immunization skewing towards IgG3, and MP is again the antigen-specific B cells, and this population again was missing in the knockout mice. We also measured another isotype that is IgM and found similar results. So the antigen-specific B cells that lack PRMT1 was very decreased if compared with wild type. And I also had a control not immunized mouse because IgM is very sticky, and to show that there's a kind of high background, and some of the cells here are due to background staining level rather than a real immune response. And again, so this is gated on CD138B220 loss, so that represent the antibody secreting cells population. And we had measured the proportion of the MP positive, so the antigen specific positive plasma cells. And as you can see, wild type had already about 20% of the antibody secreting cells were specific for the antigen that we have delivered, and very low proportion was found in the knockout mice. And again, this is just an absolute cell number of the antigen-specific antibody secreting cells that we have found in these mice. And of course, the difference is significant. So what I show you so far is that if a B cell does not have PRMT1, as incapable to generate a germinal center reaction, so there is no germinal center as well as no plasma cells that are antigen specific. So, but with the experiments I show you so far, we didn't know, so we could clearly say that one of the problems of the PRMT1 knockout B cells was to initiate a germinal center reaction, but we didn't know if in the presence of an already established germinal center, the B cell lacking PRMT1 could eventually participate. So to answer this question, we had generate bone marrow chimeras. So this is the strategy, how the experiment was set up. So the recipient mice were LY 5.1 and 0.2, and we had generate two different groups. So the group one is in principle like a control group that contained 50% of the bone marrow derived from wild type and expressed LY 5.1. The other 50% of the bone marrow derived from wild type that express the CD23 CRE, and they can be recognized with LY 5.2 marker. The group 2 include, again, 50% of the bone marrow cells are wild type, again, LY 5.1, and the rest of the 50% of the bone marrow cells derive from our flox mice that specifically express the CD23 CRE. And again, those cells can be recognized by the LY 5.2, with LY 5.2 marker. So we did repeat the immunization. So what I won't show you today, but the reconstitution was normal. The compartment wasn't showing any specific, the different groups didn't show any specific phenotypes, but I won't show you for lack of time. And after about 8 weeks of reconstitution, we immunized the mice with, again, MPKLH in alum and measure the level of germinal center in the spleen after 7 days of antigen delivery. So as you can see from here, this is group 1 and that's group 2. So this is the germinal center. And both groups, of course, had mounted a proper immune response, and they had similar proportion of germinal center B cells. However, if you now subdivide this germinal center into the two compartments, so the LY 5.1 and LY 5.2, you can clearly see that in the control group, both had participate equally. I forgot the proportion, but it's 50-50. And this second group, you can clearly see that this is the wild type, so LY 5.1, and this is the knockout. And there is almost no cells that could participate to this already established GC. And this is the quantification. So the overall absolute cell number of germinal center B cell in these two different groups. And this is the contribution of each group. So the group 1, LY 5.1 and 0.2, is almost 50-50. And this is the group 2, in which you can clearly see that only the wild type cell generate germinal center. The last question that we wanted to address was the relation of PRMT1 in a memory response. To do so, we again do bone marrow reconstitution, but this time we took advantage of another system. So as you can see from here, this is group 1, again, the control group. So this time, the wild-type mice express ERT2 CRE, that is an inducible CRE that can be induced by tamoxifen treatment. And group 2 contain, again, our the flox allele in the presence of the inducible ERT2 CRE. Again, for lack of time, so the reconstitution was normal. There was no difference between the groups. And we, after about 8 weeks of reconstitution, we had immunized the mice with, again, the same stimuli, MPKLH in alum. And after 14 days, we had measured the level of immunoglobulin to make sure that all the mice had mount a proper immune response. And I just want to remind you that at this stage, both groups should be indistinguishable, so they all still express PRMT1. And then we had waited about 11 weeks, so they were left there for 11 weeks, and at this stage we did deliver 3 times the tamoxifen, and this treatment, of course, allowed us to delete PRMT1 in all the cells. And then about a few days later, at week 12, we did give NPKLH again. We boost the mice with NPKLH, but this time without alum. And then we had monitor a very early time point that make us sure that what we've seen is due to a memory response and not a primary immune response. And at this stage, we did analyze the spleen and the bone marrow. So here is the spleen. So again, the group 1, the control group, just I want you to focus the attention on the antigen-specific cells, activated cells. You can see they respond properly, but this population was dramatically impaired in the mice in which we had induced PRMT1 deletion. And again, we had measured the germinal center that was normal in the group 1 and wasn't anymore present in our group that deleted PRMT1, suggesting that PRMT1 is required for a memory immune response. And then the last thing we have done was to measure the level of antibody-secreting cells in the spleen and in the bone marrow by ELISpot. This is just a quantification. And this is a logarithmic scale, so the difference between the wild type and the group 1 and group 2 is significant. So as you can see, apparently in the spleen as well as in the bone marrow, PRMT1 probably play a role in the antibody-secreting cell survival. So what I've shown you today is that that PRMT1 is required for germinal center B cells generation. Indeed, if you delete PRMT1, you cannot generate or participate to germinal center. And the same is true if you delete PRMT1 later on, you can also not establish memory B cells, and you also interfere with the survival of the plasma cells. So to conclude this second part, what I show you today is that PRMT1 is required for germinal center generation and maintenance, and is essential for a normal antibody-secreting cell formation. And also, PRMT1 is critical for a T-independent immune response, and is clearly essential to mount a memory immune response. But of course, what I show you today, it's only on B cells and methylation, and my question is, why is this important? So, recently, has been shown that PRMT1 is aberrantly expressed, so is upregulated in ALL. So has relevance also in other systems that are not B cells. It would be interesting to see what's the PRMT1 expression and regulation in Burkitt lymphoma using ImmuMIC system, and also what's the role of PRMT1 in autoimmunity. Indeed, with this last point, I just want to conclude the talk by showing you one results that we have recently obtained. This is the scheme of the BCR receptor, and this is the SAR kinase I've talked to you before. So when you delete this kinase, you have in the mice an autoimmunity model that is the SLE model. What we had found so far was that if you compare the level of overall arginine methylation between wild type and knockout B cell, you can clearly see that in the knockout lean knockout B cells, you have a decrease in the overall arginine methylation, suggesting that methylation might play a role in this autoimmune disease. But we still have to investigate the reason for it. So finally, I arrived to my acknowledgement. So I want to thank my boss to allow me to bring in the mice and to be always so supportive. And of course, Stephen Richard, that is the person that generated the conditional allele of PRMT1. A special thanks to Amanda and Christy, that are two amazing arrays in our lab. And seriously, this project wouldn't be possible without their help. They're always there to help, and they're amazing. So Gabriel Bells to help with the flu experiment. Vanessa to had given to me the human B cells, and Najwa and James that had given to me the QVD to do my experiment. And of course, not last but not least, the DT's angels. So all the members of the lab, so Kim, Ingela, Dimitra, Catherine, Dana, Jessica, and the other angels that are not like our boys' angels, Ivan and Michael. And of course, hopefully they're not offended, and also mouse facilities, specifically Lauren and Mel, that they always do an amazing job with the mice, the fax facility, and the founding. I can't pronounce that, so this is a German fellowship and NHMRC. Thank you for your attention.
You showed some fascinating data showing a drop in level of several of the pro-survival family members. But I wondered whether you thought this reflected them being substrates of the enzyme, that is, being methylated normally, or whether this could be an indirect effect on some upstream signaling, for example.
Unfortunately, I don't have the answer for it, but what I can tell you is I think is related to possibly post-translational modification, because the protein of those, the level of the protein of those molecules are different upon activation, but if you now measure the RNA level of the protein, they are not. So we did also some RNA microarray and did not find any difference in terms of RNA levels. I don't know if it's a direct or indirect effect of which signaling is going on inside the knockout B cell. I would like to find that. I did check the sequence of MCL1 so far, and there are some interesting arginine that could maybe be methylated, but I haven't had the time to investigate these. This is, of course, would be very good.
So you've shown that the B cell receptor is arginine methylated.
Yes.
And you've shown this immense effect of PRMT1 deletion on B cell function. But do the two connect, or are there potentially other targets that are important in the B cells, and how would you assess that if you don't know?
I do believe they are connected, but definitely there are many substrates that are methylated by PRMT1 within the B cells. And indeed, if you remember one of my first slides that I show you, if you compare the overall arginine methylation in wild-type B cells and knockout B cells, there are many substrates that are missing in the knockout B cells, suggesting that many substrates are methylated by PRMT1 in the B cells. But unfortunately, this is like what I would like to do next, to investigate which are the substrates. I don't have an answer because I think Ig alpha methylation plays a role, but is not the sole answer of this dramatic phenotype, unfortunately. Yeah.
So can you mutate that arginine and IG alpha.
Yeah.
So, and it abuts the ITAM really closely.
Yeah.
Do you know if it plays a role in SH2 binding?
Yes, it does. Yes, I didn't show you. We published that. So if you have a point mutation into Ig alpha, you have so SYK is differentially recruited to the BCR receptor. So methylation in the Ig alpha has a negative rule. So if you abolish methylation, you can recruit more SYK upon activation. And also we have found that if you mutate this arginine, so just a point mutation arginine to lysine, you also interfere with B cell development. So yes, it does play a role.
A related question. In the naive B cells where the enzyme expression is low, is Ig alpha methylated from previously, or is it a feedback sort of?
No, it's not methylated. So the sole methyltransferase is PRMT1.
No, but in
Sorry.
wild type.
Sorry. Yeah, it does. It is methylated. Sorry, I understood in the knockout. Yes, it is methylated. I didn't show, but we did measure the level of methylation in different subsets, and it is methylated. Sorry.
Okay, thank you very much.